There we go. Explain for us glycolysis and Krebs cycle. So today I can do glycolysis and another day I can explain Krebs cycle. So glycolysis, the word glycolysis is from two words, glyco meaning glucose and lysis meaning breakdown. So we can simply say glycolysis is a breakdown of glucose to release energy. And this process occurs in the cytoplasm of the cell. Now, glycolysis starts with the glucose phosphorylation, or what we call glucose activation, or simply attaching a phosphate to glucose. Now, we have adenosine triphosphate being reduced to adenosine diphosphate, meaning that it has lost one phosphate which is attached to glucose, and the resulting molecule is glucose 6-phosphate. This means that the phosphate is attached at the sixth carbon of glucose, and this process is catalyzed by an enzyme called exokinase. Now, glucose 6-phosphate is isomerized to fructose 6-phosphate. Remember, fructose and glucose are isomers. So, glucose 6-phosphate is isomerized to fructose 6-phosphate and is catalyzed by an enzyme called phosphoglucose isomerase. Now, again, we are attaching another phosphate to fructose 6-phosphate. Now, this phosphate is as the sixth carbon. We add another phosphate which will be attached to the first carbon, so the resulting compound will be fructose 1,6-biphosphate. So you can see ATP is reduced to ADP. It has lost one phosphate to this compound, and this process is catalyzed by an enzyme called phosphofructokinase 1. Now from there we have we do what we call the cleavaging, or what happens is cleavaging or cutting into two. Now, fructose is a 6-carbon compound. Now, what happens, it is divided into two. That's in simple words. So what we'll have a 3-carbon compound and another 3-carbon compound. This process is catalyzed by an enzyme called aldolase. Now, the two compounds, what we have is what we call dihydroxyacetone phosphate, and another one is glyceraldehyde 3-phosphate. The two of them are isomers. So this one can change to this one can change to this. So in glycolysis, this dihydroxyacetone phosphate will change to glyceraldehyde 3-phosphate, meaning that now there will be two molecules of this. Now, because the two can convert from this to this and this to that, it is catalyzed by an enzyme called triphosphate isomerase. Now we have glyceraldehyde 3-phosphate. Now, NAD is reduced to NADH, and also there is addition of phosphate group to the molecule. So what we resort to from glyceraldehyde 3-phosphate is what you call 1,3-biphosphoglycerate. And this process is catalyzed by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Now, from here now, we are not spending energy again. Now we are producing energy. So now you can see, in simple terms, you can say we are removing one phosphate. Remember, we have two phosphates at carbon 1 and at carbon 3. Now we remove one phosphate to produce ATP. So we have adenosine diphosphate changing to adenosine triphosphate, and because we have two molecules, then we will produce two molecules of ATP. Now one phosphate which was at the carbon 1 is removed to produce this ATP, and so we remain with a compound called 3-phosphoglycerate. From here now, what happens, the phosphate is attached at the third carbon. The next process is moving that phosphate from the third carbon to the second carbon. So what we resort to is 2-phosphoglycerate. And this reaction is catalyzed by an enzyme called phosphoglyceromutase. Now we have our 2-phosphoglycerate. Now this 2-phosphoglycerate undergoes what we call dehydration or removal of water, which is catalyzed by an enzyme called enolase, and the resulting compound is phosphoenol pyruvate, which are also two molecules. Now, phosphoenol pyruvate is converted to pyruvate, whereby we have the production of the second molecule of ATP, which is catalyzed by pyruvate kinase, and the resulting compound is pyruvate, which is actually the end product of glycolysis. So at the end of that, you see we have two molecules of ATP here, and then the other process, we had production of ATP, two molecules of ATP here. So when you look at the end products of glycolysis, we have pyruvate, which is the last step, And then we have ATP, we have water, and we have NADH. Remember, ATP are 4 molecules produced, and we had spent 2 molecules in the glucose phosphorylation, and also we had spent another ATP here. So 2 molecules of ATP were spent. So at the end of it all, we will have 2 molecules produced after like paying what you had used. Assuming you had 2 shillings, you spent, when you make 4 shillings, you return the 2 shillings, and then you remain with a profit of 2 shillings. So we have pyruvate, ATP, water, and NADH. Now this pyruvate is what is used in the next step, which is the next process or pathway, that is the Krebs cycle, because in the Krebs cycle now pyruvate will undergo oxidative decarboxylation so that it can be used to produce a molecule which is acetyl coenzyme A, which enters the tricarboxylic acid. But for today, that is glycolysis. Thank you.